Cellular Biology and Immunology. The May-June 2024 issue contains a special feature aimed at creating visibility and amplifying discussions on disability inclusion in science. The Monash Sensory Science Program has transformed the accessibility for those with blindness, low vision, and diverse needs (BLVDN) to experience biomedical data visualization through the form of multisensory scientific communication. This special feature aims to inform the inclusive teaching of immunology and raise discussions of how to improve access to all within our scientific institutions. The articles in this special feature were invited from researchers involved in the 2023 Autoimmunity Monash Sensory Science Exhibition, in which they discussed the novel artwork, models, and displays designed to improve the scientific understanding and teaching of complex autoimmune diseases, including rheumatoid arthritis, lupus, celiac disease, psoriasis, and type 1 diabetes, immunology, and cell biology. Thanks the coordinator of this special feature, Jessica Porger, for her planning and input. In our last issue of 2023, we celebrated 100 years of publication. ICB is no stranger to change, but this publication landscape is arguably changing faster right now than at any point in our history. We are committed to being at the forefront of positive author-centric improvements to the dissemination and publication of immunology, so we celebrated our birthday with an editorial workshop on the future of ICB. Our publication ethos. ICB is dedicated to identifying and disseminating quality immunology research. Over the past 100 years, we have published major conceptual shifts and technological advances in immunology. We aim to serve the community by focusing on the quality of the questions asked and the robustness of the conclusions. This year, ICB is adding a further commitment to this ethos through implementing scoop protection. Under this new policy, ICB will remain committed to the editorial decision on novelty and importance at the point of submission. Changes to the peer review process. The most important function of a journal is quality control. ICB's headline rejection rate is 70%, with an average time to rejection of 2 days. Of manuscripts sent for review, the majority of papers, 81%, are accepted in 70 days. ICB has embraced 2 new peer review approaches in 2024. The first is expedited review. Under this process, an author can transfer a manuscript cross-platform, including both the reviews and a rebuttal proposal. These expedited manuscripts get a decision without additional reviewers in an average of 12 days. The second new approach is ICB joining the Review Commons family of journals. Under the Review Commons approach, articles are submitted to the platform, undergo extensive journal-agnostic peer review, and then the authors decide on which partnered journal to transfer the article and review to. Transformative agreements and open access. As a hybrid journal, we offer subscription-based and open access publications. In the last few years, transformational agreements have allowed for free open access publication. Currently, these agreements cover most researchers in Europe, Canada, Australia, and New Zealand, and increasing numbers globally. Immunology Futures. ICB's mission is to contribute to the immunology community. As part of this mission, we use our platform to create a resource for career development. Immunology Futures is a new and vibrant resource for the community. The section includes articles on career advice, highlights diverse pathways to success, profiles immunology research hubs, and discusses future challenges for the field. Openness to ideas. ICB is a community journal run by scientists for scientists. We have a 100-year reputation to uphold, but we are also not afraid to improve our practices. We focus on changes that improve the author experience while keeping intact our reputation for integrity and quality reviewing. The editorial team at ICB keeps track of innovation in publishing, however, it is always open to new ideas directly from the community. There are 2 fundamental classifications of cells, prokaryotic and eukaryotic. Prokaryotic cells are distinguished from eukaryotic cells by the absence of a cell nucleus or other membrane-bound organelle. Prokaryotic cells are much smaller than eukaryotic cells, making them the smallest form of life. Prokaryotic cells include bacteria and archaea and lack an enclosed cell nucleus. Eukaryotic cells are found in plants, animals, fungi, and protists. They range from 10 to 100 millimeters in diameter, and their DNA is contained within a membrane-bound nucleus. Eukaryotes are organisms containing eukaryotic cells. The four eukaryotic kingdoms are Animalia, Planta, Fungi, and Protista. They both reproduce through binary fission. Bacteria, the most prominent type, had several different shapes, although most are spherical or rod-shaped. Bacteria can be classed as either gram-positive or gram-negative depending on the cell wall composition. Gram-positive bacteria have a thicker peptidoglycan layer than gram-negative bacteria. Bacterial structural features include a flagellum that helps the cell to move, ribosomes for the translation of RNA to protein, and a nucleoid that holds all the genetic material in a circular structure. Many processes occur in prokaryotic cells that allow them to survive. In prokaryotes, mRNA synthesis is initiated at a promoter sequence on the DNA template comprising two consensus sequences that recruit RNA polymerase. The prokaryotic polymerase consists of a core enzyme of 4 protein subunits and is protein that assists only with initiation. For instance, in a process termed conjugation, the fertility factor allows the bacteria to possess a pilus, which allows it to transmit DNA to another bacteria that lacks the F factor, permitting the transmittance of resistance, allowing it to survive in certain environments. Nucleus. The nucleus of the cell functions as the genome and genetic information storage for the cell, containing all the DNA organized in the form of chromosomes. It is surrounded by a nuclear envelope, which includes nuclear pores allowing for the transportation of proteins between the inside and outside of the nucleus. This is also the site for the replication of DNA, as well as the transcription of DNA to RNA. Afterward, the RNA is modified and transported out to the cytosol to be translated to protein. Nucleolus. This structure is within the nucleus, usually dense and spherical. It is the site of ribosomal RNA or RNA synthesis, which is needed for ribosomal assembly. Endoplasmic reticulum, ER. This functions to synthesize, store, and secrete proteins to the Golgi apparatus. Structurally, the endoplasmic reticulum is a network of membranes found throughout the cell and connected to the nucleus. The membranes are slightly different from cell to cell, and a cell's function determines the size and structure of the ER. Mitochondria, commonly known as the powerhouse of the cell, is a double membrane-bound cell organelle. This functions for the production of energy, or ATP, within the cell. Specifically, this is the place where the Krebs cycle or TCA cycle for the production of NADH and FADH occurs. Afterwards, these products are used within the electron transport chain, ETC, and oxidative phosphorylation for the final production of ATP. Golgi apparatus. This functions to further process, package, and secrete the proteins to their destination. The proteins contain a signal sequence that allows the Golgi apparatus to recognize and direct it to the correct place. Golgi apparatus also produces glycoproteins and glycolipids. Lysosome. The lysosome functions to degrade material brought in from the outside of the cell or old organelles. This contains many acid hydrolyses, proteases, nucleases, and lipases, which break down the various molecules. Autophagy is the process of degradation through lysosomes, which occurs when a vesicle buds off from the ER and engulfs the material, then attaches and fuses with the lysosome to allow the material to be degraded. What do you think of our video? Let me know in the comment section below. Before you go, please hit the like button and subscribe to my channel. Thanks for watching.